We have we have met these fine folks in the past, and they're wonderful people who they're here to talk to us today about some research that's happening at the Michael J. Fox Foundation. They'll talk about some new developments in Parkinson's medications. They'll talk about some lifestyle things related to Parkinson's, and they'll also save some time at the end for you to ask questions. For those of you who do not know what Power Over Parkinson's is, we're actually a group located in Monterey. We're a nonprofit that our goal is really to help provide information and services to people with Parkinson's to help improve their lives or enhance it in any way that we can. So today, this is the first of our community speaker series where our goal is to, find people in the area of Parkinson's research or Parkinson's expertise to just kind of bring you any new information, keep you informed, and keep you engaged. So today we have Jennifer Rydell from the Michael J. Fox Foundation, who is a Senior Associate Director of Advancement, and Susanna Watch is an Advancement Officer at the Michael J. Fox Foundation. And they work with community members and supporters of the foundation in the Western US and Western Canada, and they're really excited to be here with all of us today. Normally they would be doing this in person, and so they're both in New York, and so they don't get to be here in California with us. But I'm going to welcome Susanna in, and the rest of the show is going to be yours for today. And again, at the end, there will be time for you to ask questions. So if you do have questions that pop up during the presentation, feel free to drop them in the chat so you don't forget, or write it down on a piece of paper in front of you so that you can ask those questions at the end. And so with that, I'm going to turn it over to Susanna.
Great. Thank you, Maria. Like Maria mentioned, I'm here along with my colleague, Jennifer Rydell from the Michael J. Fox Foundation, and we're so glad to be here with you today. We normally, again, would be in person, but this is the best we can do for now, and I'm so glad to be with you all. And I do want to first just acknowledge that we are in a virtual setting, and as Maria said, ideally, this could be more of a conversation, but just for ease of focus for everyone, I do want to encourage you to drop any questions you may have in the chat. You'll see that Jennifer has started to activate the chat, so please feel free to chat in there, ask any questions, comments, and Jennifer will continue to add helpful links as we go through this presentation. So I'm going to share my screen. I do have a PowerPoint presentation that will accompany my update. And so that everyone knows, this will be recorded. So if you miss anything, you want to try and remember something, don't worry about it. We'll send out a recording and a copy of these slides so you have access to them as you want them. So today, first we'll start, this will be a research update, but I'll start a little bit with a background of the foundation. Then we'll dive into the meat of the presentation, which is our research update. And lastly, I want to discuss ways that you all can get involved with the foundation and with the Parkinson's community in general. And you can stay connected with us as parts of the community. Maybe you're a person with Parkinson's, a family member, or a supporter. So I like to start by walking through our slogan, which was new as of 2019, and that's "Here until Parkinson's isn't." And I think that it really embodies the foundation's goal overall, which is that we are here for everyone in the Parkinson's community to find a cure, and then we'll close our doors. That is our goal, and we are working every day to improve the daily lives of people with Parkinson's by improving symptomatic treatments while we work towards a cure. So you'll see our mission here on the screen. I'm not going to read everything on the slide, just, I'm not here to recite to you, but I do want to just point out a few things. We are, of course, founded by the actor Michael J. Fox back in 2000. And, along with that, we have been a patient-centered organization since the beginning. Michael is, of course, a prolific actor, beloved by many, but he is a person with Parkinson's. And so he brings that voice to our foundation. We're a pretty lean team of about 160 folks. We're based in New York City and we do not have chapters. This helps us stay very efficient with our donor dollars. And about 88 cents of every dollar raised goes straight to research funding. And we can do this because we don't have brick and mortar locations all over the country. And we're really focused on getting donor dollars from our foundation out to our grantees. To that end, we have about 100,000 donors, who supported our mission in 2020. And since our inception, we have sent out more than $1 billion to researchers. In California alone, in 2020, we funded $24.2 million in grants, which is awesome. California, you guys are usually at the top of our list, and we're glad to be able to fund so much exciting research in your backyard. You'll see here that actually about a third of our funding gets disseminated outside of the US. So we're really looking to fund research that's promising and novel wherever it may be, even if it's not here in the US. And we convene over 400 researchers each year for them to share their ideas and information to make sure that we're collaborating as a larger Parkinson's community. So what makes us unique? I want to dive into our business model a bit just to show you how we work and how we might be different than other organizations you've learned about or heard from in the past. So like I said before, patients are truly at the center of everything we do here at the foundation, and they are really what guides us through our granting process. So you'll see at the bottom here, we have this little cycle where donors are at one end, we're in the middle, and awardees are at the other. And throughout this whole process, we're taking into account the needs and wants of our patient community. They're helping us guide our research in many ways. We also deploy our funding to researchers within just months of receiving donations. That average is about 6 months, in the door to us, out the door to researchers. Our research team is on staff, and it's a group of 13 PhDs, as well as one doctor who's a movement disorder specialist, which is a neurologist with special training in movement disorders like Parkinson's, who work alongside 12 business strategists to help us make strategic decisions about where we should be funding research, looking at what's promising, where it is, how efficient we can be with our dollars. Our grants are also milestone driven with quarterly assessments, which gives us the ability to be very nimble with our research dollars. So once you're granted a grant from the Michael J. Fox Foundation, you have to check in with us on a quarterly basis to provide progress updates. This allows us to ensure that your research is going well, and we can offer information or advice as you're hitting roadblocks. If a researcher does hit a roadblock that has kind of brought their research to a halt, we can then redirect that funding elsewhere to make sure that we're being most efficient again with those dollars and getting them where they can go farthest. To that end, unexpected results are still valuable. And so because of that, we make sure that our grant making process has a stipulation that collaboration is required. So once you get a grant from us, you must share your research and your findings. So even those negative results can be shared with others who can learn from that knowledge and grow from it. So very important to us that research is shared, which is not always the norm in the research field. So here on this next slide, you'll see our research roadmap, which is really how we make those strategic decisions around our grant making. It helps us just stay prioritized in the right direction with our funding. So we see 3 words here with one in the middle: define, treat, and measure. And then tools and resources is at the center of everything we do. So define meaning understanding the underlying causes of PD, its progression, and assessing the efficacy of treatments. So defining the biology of PD. Treating, meaning to develop treatments to slow, stop, reverse the progression of Parkinson's, as well as treat the everyday symptoms. And then measure, looking at how can we diagnose Parkinson's and how can we track progress? How can we measure what's happening in the body? A big part of this is discovering a biomarker, which I'll discuss later. So just lock that word in the back of your heads. And then at the center of this all is tools and resources. So what tools can we develop and what resources can we share that can help everyone in the Parkinson's research field move faster? So this slide, I wanted to explain to you all kind of where we positioned ourselves in the drug development pipeline in order to be most efficient and to have the largest impact on the larger field. So drug development is a pretty long process that historically has taken anywhere from 15 to 30 years to get from idea to pharmacy shelf, and about $1 billion, billion with a B. So it starts in discovery, which is looking at basic biology, looking at cells or proteins or pathways, and not focusing on a specific disease, just trying to understand biology. This part of research is usually funded by the NIH in the US and other governments elsewhere. Next, we move into target validation, which is where the Michael J. Fox Foundation positions itself to be most efficient and to help move things along. This is kind of the beginning of our timeline. Target validation is looking to discover if a target that we discovered in this discovery phase is actually applicable to a specific disease, in our case, of course, Parkinson's. Then we move into translation, which is translating that target into something that's druggable. So looking at a small molecule that can be taken orally, perhaps you're looking at a vaccine, as we all know is kind of top of mind for everyone today. Translating that idea into something that we can administer to people with Parkinson's. Then we move into human clinical trials, which is split up into 3 phases, as you may know, phase 1, phase 2, and phase 3. Phase 1, looking at a small group of healthy volunteers to just test mostly for safety. Phase 2, looking at safety and efficacy in a small group of people with the disease in question, in this case, again, Parkinson's. And then phase 3 is a large-scale, longer-term study of long-term efficacy and safety of the drug in question to see if it's really doing what we want it to and if it's safe for the general public. Now, in phase 3 of human clinical studies, things can get very expensive. It's a huge undertaking for the pharmaceutical company or whomever is leading the trial. And so at this point, we look to biotechnology companies, pharmaceutical companies, or venture capitalists to fund this research. But because we at the foundation have positioned ourselves before that phase 1 or phase 2 or phase 3, we have essentially de-risked the progress at hand. So we've shown these larger companies with more at stake, they're for-profit companies, that it's worthwhile for them to investing in this specific drug because they're likely to have a return on their investment. So they're looking for that reliable return. And by having the foundation fund those earlier phase trials, we've taken a lot of the risk out of funding that later stage, more expensive research. And then finally, the drug will move into regulatory approval where it's approved by, in the US, the FDA. So because we've positioned ourselves kind of in the middle of this pipeline, we're not afraid to fund that more novel or unknown research because our goal is really to get things through to phase 3, where we can get larger companies to take it on. So by funding all different kinds of ideas, or in our words, having multiple shots on goal, we can really make sure that we're leaving no stone unturned. Now, I do want to point out that I said earlier that most studies typically in the past have taken about 15 to 30 years to get through this pipeline. With the help of the Michael J. Fox Foundation, we have seen some drugs come to market in as soon as 12 to 15 years, and some even faster, which we're so pleased about. So now that we've discussed the foundation a little bit and how we stand in research, I do want to start the research update. So we break things down into 3 different priority areas, which is how we'll discuss them today. We'll start with symptomatic treatments, which is treatments that look to address the motor and non-motor symptoms in Parkinson's, and also other emerging targets. Then we'll discuss disease-modifying treatments and research that we're looking at, which is looking to slow or stop the progression of Parkinson's disease, or what some people may consider a cure. The areas of interest that we'll discuss today are genetics, including the genetic mutations of LRRK2 and GBA, as well as alpha-synuclein and some other emerging targets. And then finally, we'll finish with field-wide challenges, which basically is us looking at what we can do to speed up the field. And this includes biomarkers, increased clinical trial recruitment, and research tools. So we'll start today with that symptomatic treatment bucket. So on this slide, you'll see that there have been 39 treatments approved for symptoms of Parkinson's disease since about 1954, which is when the first therapy for Parkinson's was approved. You'll notice that back, in the 1954 to about 2015 part of the scale, most drugs are color coded in that orange color, which means they address general symptoms, usually dopamine replacement therapy to help with that tremor or rigidity. However, we know that there are a lot more symptoms that can comprise Parkinson's disease than just motor symptoms of tremor. We like to say that if you've met one person with Parkinson's, you've just met one person with Parkinson's, because everyone's journey is so different and can make up so many different symptoms. So you'll see in these more recent years, the colors of these different drugs have changed, showing that they're addressing a lot of different symptoms aside from just the general motor. We have things addressing orthostatic hypertension, psychosis, increased drooling, and others. So I'm glad to see that in recent years, we've seen a lot of progress in addressing different symptoms. In fact, in 2020, we saw 3 new therapies approved just in the year. And as we know, it was a tough year. So for us to see 3 new therapies approved was wonderful. 2 of them were addressing off times in Parkinson's, which is when you may begin to feel your medication wear off, but it's not time for your next dose. So these drugs can help mitigate the amount of off time you feel during the day. And then we also saw 1 deep brain stimulation device approved in 2020. I want to note that one interesting study that we're funding just up the road from you all in this bucket of symptomatic treatments is happening at UCSF, where researchers are looking to improve gait and balance in people with Parkinson's by adding on to already scheduled deep brain stimulation procedures to see if they can activate different parts of the brain during different parts of the walking cycle to see if that improves the user's gait and balance. So no one's getting an extra surgery because they're already getting their deep brain stimulation, but the researchers are looking to see if they can start activating a different part of the brain called the striatum to see if that will improve someone's step cycle. So very interesting research, and I do hope to keep you updated on what happens with that. We just funded it at the end of last year. I want to share kind of a case study of a symptomatic treatment that we funded in early stages and that now is on the market. So this is called Inbrija, and Inbrija is taken as kind of like an inhaler, and it's used to mitigate off times, which I kind of explained before as that onset of symptoms before it's time for your next dose. So Inbrija works, you use it as a rescue inhaler. It's used as soon as you start feeling those off times come on. You can just take a puff of the inhaler and it works quickly. It's absorbed through the cheeks up into the brain and can deliver that extra dose of dopamine that you need to get you to your next dose. So in 2011, we funded a phase 1 study of Inbrija, which was about $1 million. And then from 2011 to 2017, we helped connect participants with this study. In 2013, we then gave follow-on funding, another about $1 million to fund that phase 2 study. And as I mentioned before, after phase 2 is where it starts to get very costly. And because we de-risked this study, a large biotechnology company called Civitas was able to raise another $93 million to continue the drug development. Again, another great step in the de-risking process is that Accorda saw the promise of this therapy and acquired Civitas, which led them to more follow-on support to continue through and to the goal line to getting this approved by the FDA in 2018. So in just about 7 or 8 years from the phase 1 study to FDA approval, we were able to see a great new therapy for off times come to market that has really changed how people are able to live their everyday lives, knowing that they have this inhaler in their back pocket if they need it when they feel an off time coming on, so they can go to lunch with friends or take their dog for a walk, which is something maybe they may not have been able to do before because of those off times. I want to note that we actually saw another case study of the foundation's early funding come to market last year with a drug called Kinmobi, which is also a rescue therapy for off times. It was approved just about this time last year as an under the tongue apomorphine strip. So it looks something like a Listerine strip. And it delivers apomorphine, which helps promote more use of dopamine in the brain. You may have heard of it before in the form of apokin, which is another rescue therapy that was only available as injection. So having apomorphine available in an under the tongue strip makes it much less invasive and painful for someone with Parkinson's to use it. And it also gives people with Parkinson's more than one option when it comes to rescue therapies. And, as I said before, not everyone with Parkinson's has the same journey of symptoms. And so it's important to have options for people to explore different ways to address their symptoms. So we're very proud to have both Inbrija and Kinmobi on the market to help people mitigate their off symptoms. But there are a lot more ways to address symptoms of Parkinson's than pharmacological interventions. So we also explore these innovative ways to address unmet needs through non-pharmacological interventions, or in other words, not drugs. So in this case, we're looking at a lot of wearable devices, which is very exciting because these can take only about 5 years to come to market instead of that 12 to 15-year timeline, because we're not injecting anything into the body, we're not ingesting anything, so it can happen a lot faster. These wearables can also get scientists from many other fields involved in Parkinson's research, which is so exciting for us to see that we're motivating engineers and physicists and designers to come into the Parkinson's field. So just to walk you through these quickly, these were all funded back in 2018 with a $3 million grant. So we're hoping to see more news from most of these in the next year and a half. We have these vibrating socks on the left, which can help with shuffling and freezing of gait, which is a symptom some of you may be familiar with. But it's a sock that as the user begins to freeze or shuffle, it vibrates, which then sends a signal up to the brain to remind the user to begin to pick up that regular pace of walking again. In the middle is a waist-worn airbag, which works to help mitigate injury from fall. Some people can lose their balance a little bit, and this can help with that. So if the user begins to lean to one side, the waist-worn airbag will inflate just a bit to pick them back up, and then they can continue on. And in the case or in the event of a fall, the airbag will inflate all the way and help prevent any serious injury. So you'll see that it's discreetly worn just over the underwear and underneath pants. So it's not some big bulky airbag that might be, a little embarrassing to wear in public. And a big part of this was that we got our patient council, which is a group of patients who are partners to the foundation, involved with the development of this product. And a company came to us with basically a cage to be worn as a protection. And our patient council said, we understand why you want to make this product for us, but we're not going to wear that. So, try again and think of something that actually is practical for everyday use. And then, of course, the waist-worn airbag was born, which is great. And then on the right, we have an assistive walking device, which is actually made in collaboration with Honda, which is very cool. And this is used if people are having more severe mobility issues, and it helps us just return that full range of motion. So for people who may not be able to walk, longer stretches of time or around the home, this can help them return to that mobile ability that they had before. So next, we're going to get into really the meat of the research update, which is therapies to slow or stop the progression of Parkinson's, or again, in other words, some may consider that a cure. So this is split up into 3 different sections. We'll discuss alpha-synuclein, then genetics like GBA and LRRK2, and then other therapies that we're investigating. So to ease us into this, I do want to start with just a little analogy that might help us illustrate why this is different than a treatment for symptoms of Parkinson's. And that's when we think about Parkinson's, we mostly think about the loss of dopamine, right? So we're looking at dopamine replacement therapies when we think about most symptomatic treatments for Parkinson's disease. If we think of our brain as a fishbowl, we can think of dopamine as the fish, but there's other things involved in the fishbowl. There's water, food, some rocks at the bottom, maybe a decorative treasure chest or something of the sort. And if our goldfish begin to die, we can easily take them out and replace them. That's what we're doing with that dopamine replacement therapy. We're replacing the goldfish. But why are they dying? Is it the dirty water? Is it the food's no good for them? Are the rocks not working with them? What is causing the toxic environment that's at the root of our dopamine cell death? So that's where we look at these therapies to slow or stop the progression, and where alpha-synuclein really comes into play. So alpha-synuclein is a protein that everyone has. It's in our blood, it's in our skin cells, it's everywhere. But we see that in people with Parkinson's, almost everyone with Parkinson's, the alpha-synuclein protein starts to clump and misfold, creating a toxic version of the protein. These clumps are called Lewy bodies. In people with Lewy bodies, we begin to see the cells that they're in break down. Those are those dopamine cells. And so this is one of our most promising routes of figuring out how to stop or slow the progression of Parkinson's disease, because we think that if we can break down those toxic clumps or prevent them from forming in the first place, we can slow or stop that progression of Parkinson's disease. So we currently have 13 therapies across phase 1 and phase 2 trials of humans looking to break down or prevent the clumping of those Lewy bodies, that toxic alpha-synuclein, which is excellent progress. And I know that there's going to be a lot of movement in this area in the next couple of years. I'll dive in a little bit more into the mechanics of how we're looking to prevent that clumping on the next slide. But before we get there, I want to just quickly discuss genetics, which looks at GBA and LRRK2, as well as many other genetic mutations, but GBA and LRRK2 are the most common. So, about only 10% of people with Parkinson's have a known genetic mutation that has increased their risk for developing the disease. But we know that these genetic mutations can lead to changes in pathways that we see in most people with Parkinson's. So even if you don't have a genetic mutation, we often see a similar pathway leading to those clumped alpha-synuclein Lewy bodies, regardless of the genetic mutation. So while a mutation may cause it, other things can cause that change in pathway as well, like environmental factors, for example, head trauma, well water, toxins of war. There are a few others. So we know that genetics plays a strong role, as well as the environment, and it's important for us to investigate both. GBA is the first genetic mutation that I'll discuss quickly, and GBA is a gene that encodes for a GCase. A GCase is an enzyme that breaks things down. So when we see a mutation in the GBA gene, which encodes for GCase, we see that the GCase enzyme is not doing as good of a job breaking things down. That leads to an increase in that toxic alpha-synuclein again. So we're looking at, the lack of breaking down of certain proteins can slow down the lysosome, which is your garbage disposal, which can then lead it to not working as well and can lead to the accumulation again of that alpha-synuclein. So a very promising pathway for us to investigate. It's the most common genetic mutation in Parkinson's, and we have 4 therapies against that GBA mutation currently in human trials. The second genetic mutation we'll discuss today is LRRK2, which accounts for about 1 to 2% of Parkinson's cases, but we interestingly see the same pathway in many people with Parkinson's. So LRRK2 encodes for a kinase, which works as an on and off switch in the brain. Kinases in general are studied a lot by cancer researchers because that on and off pathway can lead to the development of cancer cells. Now, it doesn't in the case of Parkinson's, but it's a similar pathway. So we're lucky to have all of that cancer research kind of on our side in the background that's helped us learn so much about LRRK2 so quickly. LRRK2 also leads to the accumulation of those Lewy bodies, that misfolded alpha-synuclein, because it's on too much in Parkinson's. So it's creating too much alpha-synuclein. Because of a very generous donor, we've been able to work very quickly through the LRRK2 space. And in 2017, we saw our first trial come to human clinical testing. And now we already have 2 therapies in LRRK2 human clinical trials. So really exciting work there. And I think we'll see a lot of progress in that in the next few years. Now, of course, there are other avenues that we're investigating into how to slow or stop the progression of Parkinson's. Again, we leave no stone unturned. So some of those other therapies include looking at repurposed drugs, or looking at drugs that are already approved by the FDA for other indications and seeing if they can help improve the lives of people with Parkinson's. One great thing about looking at repurposed medications is that we can often get them through the pipeline a lot faster because they've already been tested for safety and tolerability in the general public. So we know that they're safe and they can often move through the pipeline a lot faster. Some other targets we're looking at include inflammation, mitochondria, oxidative stress, and cell repair. So lots going on there. As I promised, I did want to dive a little bit deeper into the mechanisms of how we look to break down or prevent the clumping of alpha-synuclein. So there's 4 different mechanisms that we're looking at right now. The first is translation, and this works to make less alpha-synuclein in the first place. So translation is when we move things from DNA to RNA to protein, right? So we're stopping that translation timeline. We're not letting as much alpha-synuclein protein be created. Next is clearance, which is working to improve the garbage disposal mechanism of the cell, essentially, looking at how we can encourage the body to work better through its garbage disposal process. And again, this goes back to that lysosome that I mentioned earlier and helping to make that work more efficiently. Next is immunotherapy, which is kind of the converse of clearance because we're adding an outside agent like an antibody, which might be a buzzword for a lot of us right now. We're looking at introducing an antibody to the brain that can help break down the alpha-synuclein if the garbage disposal within our own brain isn't working too well. And then last is disaggregation, which works to bind to that toxic alpha-synuclein and prevent it from misfolding. So before it can fold up, we bind to it and encourage it to fold in the right shape. One study I did want to highlight in this work is from MODAG, which is a German biotechnology company. We actually just dispersed a grant to them for $1.4 million at the end of 2020 to look at a phase 1B study of their small molecule, which has already had some great safety results. And that's looking at binding to the toxic precursor of alpha-synuclein. So like I mentioned before, it has to fold up. So we're looking at binding to it before it's unfolded. It's called a toxic oligomer, and preventing it from folding incorrectly. So we're hoping to see some great news from that in the next year or two as it works through its phase 1B trial. I want to discuss just a bit about how these genetic discoveries that we discussed before in LRRK2 and GBA, as well as others, can help the broader Parkinson's community. So as I mentioned, we believe that Parkinson's is a combination of genetics and the environment. So while some people may have a stronger genetic predisposition, we know that there are environmental factors that can often, be a turning point into the onset of Parkinson's. And we see that people who have no genetic predisposition often still exhibit the same pathway in biology in their brain that leads to these toxic clumps of alpha-synuclein. So while we can find a genetic mutation like that LRRK2 mutation, we can examine populations of people who have it and figure out the biology behind it. Then we can zoom out and take a look at that biology as it relates to other people with Parkinson's. Do we see people who have idiopathic or unknown cause Parkinson's have a similar process happening in their brains? Do they also have, a LRRK2 kinase that's extra on? Do they have that same pathway going on? And then we can decide, will the therapy help the larger PD population? And the answer so far has been yes. So we're very invested in these genetic discoveries because we know that, the Human Genome Project wasn't that long ago. So the more we can figure out about genetics and other possible links, the more we can relate it back to the larger Parkinson's community and make cures that work for everyone. So now I want to move us into our third priority area, which are those field-wide challenges, or what can help us move faster? What can we do to help the field get over those hurdles to get to the end goal? Now, the biggest challenge here is a biomarker or a measurable biological characteristic that we can use to definitively diagnose Parkinson's and track its progression. A biomarker, again, would help with diagnosis. As some of you may know, a diagnosis process for Parkinson's can be very subjective. It can require people to, see a doctor and do movements, which isn't objective. It requires, some deduction. It also requires many tests, not just one blood test, for example. It would also help us chart disease progression to see how it changes over time. For example, if we could just take a blood test and measure a change in something over time, we can see if the disease is progressing or regressing. Having a biomarker can also help us design better clinical trials, and it can help us test the impact of new therapies. We actually announced a $2 million prize for anyone who can come up with an imaging biomarker for Parkinson's just last year or 2 years ago. And we've seen some great progress from that already. In fact, we had 3 awardees, and one of them has already moved on to human clinical trials of their imaging biomarker for toxic alpha-synuclein in the brain. So we're thinking of something like a PET tracer, where you'd get an intravenous injection of this substance that would bind to the toxic alpha-synuclein in the brain. And then after a brain scan, it would light up where those clumps are. Currently, we don't have this, but AC Immune is in human clinical trials of their PET tracer testing for safety in phase 1. And we are so thrilled to see this quick progress. And, you know, this really would help turn the field into a whole new direction. Now, we're so invested in finding a biomarker for Parkinson's that we've actually created our own landmark study. It's called the Parkinson's Progression Markers Initiative, or PPMI, and currently it runs in 33 international clinical sites with 1,400 participants. We've seen this data downloaded over 5 million times, and we're actually looking to revamp the study this year. This is hot off the presses. We're expanding to 50 clinical locations in 12 countries and recruiting 4,000 new participants. So Parkinson's Progression Markers Initiative, again, or PPMI, takes biosamples from people with and without Parkinson's and tracks them over 5 to 10 years. We're looking for changes in anything that can be blood, cerebrospinal fluid, tears, skin samples, and others, DAT scans. We're looking at everything to see what changes over time, from not having a diagnosis perhaps to having a diagnosis, or as your Parkinson's progresses, what's changing? And if we can pinpoint a measurable biological sample that we can see changes over time that might predict Parkinson's, or it might, show the change over time, we can then use that as a measurable biomarker for people around the world. Our co-founder, Debbie Brooks, is actually enrolled as a control participant in this trial just to show how committed we are. And she has been giving her biosamples for the last 10 years. For this new recruitment, we're really focusing on a cohort of people with known risk factors of Parkinson's, but who do not have a diagnosis. So these are people who are children or parents or siblings of someone with Parkinson's, someone who has a known genetic mutation, who has a certain sleep disorder called REM, sleep behavior disorder, or who has degenerative hyposmia, which is loss of smell over time. So we're really focusing on looking at people who have those risk factors and seeing what changes over time. Now, lastly, I want to discuss what you all can do today to become more active members of the Parkinson's community, or perhaps get involved with our foundation. So this next slide can be a little overwhelming, and I want to just encourage everyone to take one action item away from today to see if they want to, jump on it and get started. So we'll start with exercise. This can help you manage symptoms, of course. I know Maria speaks about this a lot, and it's so important for everyone to have an exercise regimen that works for them. Again, there's no right exercise for Parkinson's, but we do find that helping with flexibility can help mitigate injury and falls. So this includes yoga and Tai Chi, maybe boxing, but we always, of course, recommend that you work with a physical therapist to build a safe regimen. Next is self-care activities, and I want everyone to give themselves a pat on the back, because being part of a group, a support group, is a great self-care activity to help you feel like you're part of a community, which of course you absolutely are, to share your experiences with others and get feedback. It's just a wonderful step to take. So congrats to you all. But of course, there are other self-care activities, which can include picking up a new hobby, getting involved with your larger community, maybe doing a creative project. So just helping yourself feel well day to day. Next is diet. And we always recommend that a healthy, balanced diet, which typically ends up falling into this Mediterranean diet of lots of fish and lean meat, and kind of cutting back on that red meat and having lots of whole, healthy fruits and vegetables can really help you feel your best every day. And we also want to note that sometimes you might want to make an adjustment to your diet to help you feel the best with your medication. So you may notice that if you eat too soon after taking your meds, they may not work as well. Thinking about timing that. Of course, talk to your doctor before you add any vitamins or supplements to your diet as well. And then lastly, building a care team can be so helpful for people with Parkinson's to make sure that they're hitting all their bases. This can include a movement disorder specialist, which we recommend that you see about once a year. This is, again, a neurologist with special training in Parkinson's disease and other movement disorders. But this can also include people like a physical therapist or a counselor or a nutritionist and others. So think about, maybe rounding out your care team if that's something that might help you. There are other ways to get involved on the research end, which I know is something that can excite many of you. The first is joining our online observational clinical study called Fox Insight. This is run by the foundation and it's completely online. You can do it from the comfort of your own home, which of course, is super relevant right now in the time of the coronavirus for us to be safe at home, but still be involved in research. So you log on quarterly and take essentially short doctor's visits where you self-report your symptoms and experiences. And this patient-reported data helps us understand what's needed from the Parkinson's community and what daily life with Parkinson's is like. Now, I'm actually enrolled as a control participant. I take the surveys every quarter, and it really is, I like the check-in, even though I don't have Parkinson's. It helps me check in with my overall health, and I can then print out my study visits every quarter and take them to my doctor. So let's say you're seeing your movement disorder specialist at the end of the year, but you can't quite recall that in the middle of the year, you were having some trouble with some symptoms. If you have these study visits you can print out and bring, you can then recall, back in June, I was discussing over Fox Insight that I was having more trouble with my rigidity than normal, for example. So having these study visits in your back pocket can be really helpful as you navigate Parkinson's disease. I will mention that we also have off-cycle surveys that come out on Fox Insight on specific topics. So recently we deployed a coronavirus survey to see how people who've had COVID has affected their Parkinson's, if at all. We've also deployed medical marijuana surveys and others to help us understand more broadly life with Parkinson's. We also do want to mention that we encourage people to participate in clinical trials if it's the right fit for them. You'll see a statistic here on the screen that though 88% of people with Parkinson's say they'll be willing to participate in a trial, not many do. And we know that having people participate in clinical trials is really what will help us move faster, because if research groups can focus more on the actual trial than on recruiting participants, they can move a lot faster. So to help people get enrolled and to find out what works for them, we created an online resource called Fox Trial Finder, which is essentially like a Match.com, but for finding clinical trials. So you can go online and type in your location, type in how far you're willing to go, and find trials in your area. So I'll note that I did a quick query this morning for within about 100 miles of the Monterey and Carmel area, and there are 23 clinical trials actively recruiting right now in your area. These are a mix of interventional and observational trials. So interventional meaning that a team will administer some kind of drug or treatment and an intervention essentially, and then evaluate the outcomes. Whereas an observational trial means the research team is simply observing without intervening at all. So Fox Trial Finder is a great place to go if you're looking to enroll in an in-person or online trial. I do want to mention that we have a lot of resources on our website for you to get more involved with the foundation and to continue to educate yourself on life with Parkinson's and different treatments. So I'll just list a few of them here, but our website really is a great resource. We have webinars every third Thursday of the month, which are live discussions with experts from all over the field on different topics. We also have podcasts, and we have an Ask the MD series with our on-staff movement disorder specialist, where she publishes blogs and videos all about different questions in Parkinson's. You can also get involved through Team Fox, which is our grassroots fundraising arm of the foundation. Say you want to honor a birthday or an anniversary, or join us for a walk or run or bike ride on behalf of the foundation, that's your place to go. And then lastly, you can get involved with advocating for Parkinson's policies through our policy arm, which is at that website there on the screen, and Jennifer will also post it. Here you can learn more about policies that are affecting people with Parkinson's and who you can contact, local elected officials and policymakers, to help encourage them to see how their policies are affecting people with Parkinson's and what political change they can make. Lastly, I want to point out that we do have a lot of published resources to help people who are living with Parkinson's or are loved ones or caregivers to people with Parkinson's. And one example of that is Parkinson's 360, which is a comprehensive guide on Parkinson's written by our movement disorder specialist and also by patients. We have a wonderful group of patients on our patient council who helped us publish a really effective guide that touches so many different parts of the lived experience with Parkinson's. So with that, I've covered a lot of information, and I haven't been able to see the chat, but I have seen that there have been some messages. And so with that, I'd love to get to some questions. Again, I discussed a lot today, but I hope that you found it helpful. And again, we will be sending all of this as a PDF and a recording. So if you missed anything throughout the conversation, not to worry. We'll have resources to everything linked, and you can go back and watch anything you missed. And if you're interested in learning more, Jennifer will post a link. We will have a survey that we'd love for you all to take if you want to help us improve these presentations, but also help us, figure out what you guys are interested in learning more about. So you can feel free to provide your contact information. We can get back to you all with answers to any one-off questions you may have that you don't want to share today. With that, we have about 10 minutes left, and I'd love to hear from you all. Jennifer, hear from you first. If anyone asked any questions in the chat, I'd love to hear those.
Yeah, absolutely. People have been loving your presentation, so a lot of compliments in the chat as well as questions. I'll go ahead and kick it off with our first question from Roger, and Roger's interested to know about focused ultrasound, and has Michael J. Fox Foundation been involved in focused ultrasound or any clinical trials surrounding that?
Hi, Roger. That's a great question. And yes, we have seen a lot of progress in focused ultrasound, especially recently. I'll point out that one of the biggest differences between focused ultrasound and kind of its sister treatment of deep brain stimulation is that focused ultrasound is a non-invasive way to target very, very specific areas of the brain, which can often help with those motor symptoms. Now, it's irreversible, whereas with deep brain stimulation, it is reversible because it's just an electrode that they can remove from the brain. So definitely pros and cons to both, which you should discuss with your movement disorder specialist, and one may be the best fit for you. We have, like I said before, seen a lot of progress in the field of focused ultrasound in the last couple years. And I actually read an interesting article that was sent to me by a community member just a couple weeks ago about looking at focused ultrasound, focusing on a different section of the brain. So if we can target different locations in the brain, we might be able to target very precise symptoms. So we're seeing a lot of movement in it. If it's something that you're thinking about right now for those motor symptoms, I'd say absolutely ask if it might be the right fit for you, but there is a lot of progress still happening in the focused ultrasound space.
That's fantastic. Our next question comes from Maria, and Maria's interested in understanding, is the Michael J. Fox Foundation funding any research on the use of cannabis? And if yes, is there any preliminary results in that area?
A very great question, and often a complicated one, because medical marijuana is not uniformly regulated across the US. So it's hard for us to study in the lab because we don't know exactly what people are using when they say they're using medical marijuana. When you buy CBD, for example, or actual THC, you might be getting different levels in different markets. It's not actually regulated by the FDA, so the level that you see on the packaging may not be exactly accurate, but we have been studying it. We actually have a researcher on our team named Andrew, who we kind of think of as our pot PD expert. He has done a lot of work in the field, and he's very up to date, and he's really keeping us with our finger on the pulse. And I mentioned before, Fox Insight had a special survey that we deployed all about medical marijuana. So this was the largest of its kind, and we've seen a lot of results from it so far. Andrew, I believe, is planning on sharing those results at a consortium coming up. But we're hoping to see a lot more soon. Unfortunately, I don't have any definitive outcomes quite yet, just because, as I mentioned, it's not that regulated, and it's also hard for researchers to get it into their lab safely, because again, it's still a Schedule I drug in a lot of places, so it's not easy to get to study for medical purposes. But that's not to say we aren't trying our very best, and we're looking into it every day. So it's definitely on our radar. And if you're thinking about using medical marijuana, I'd say, of course, discuss with your physician, but we have seen some people who've found a lot of great outcomes from it. For other people, it doesn't work so well. So it really is case by case, and feel free to explore it if it's something that you're thinking about.
That's fantastic. Our next question is regarding stem cells. And has there been any movement in the stem cell area? And if so, how has the Michael J. Fox Foundation been involved? For example, have they funded any recent studies regarding this subject area?
I feel like this question was planted because this is such a great question and very top of mind for me. We just had a big study that we funded at the end of 2020 at the University of Texas at Houston, which is their medical center, looking at mesenchymal stem cells. So these stem cells are able to travel to sites of injury or inflammation and decrease inflammation. So like I talked about with therapies to slow or stop Parkinson's, inflammation is one of those pathways that we're investigating. And if we can figure out how these mesenchymal stem cells can stop so much inflammation from happening, this might be able to help us get to the root of Parkinson's and slow down its progression. So we are absolutely funding stem cell research. It's a definitely emerging field. We're still learning about it as we go. As you may or may not know, there are lots of different kinds of stem cells and different kinds to make them, ways to make them in the lab. So moving very quickly here, but I'm very excited about this research at the University of Texas. It's a human clinical study, so we're hoping to see some great things from them in the next couple of years as they're able to recruit and get that study off the ground.
That's fantastic and really encouraging. Our last question is in regards to Michael himself, and just a question about, is Michael actually involved in the foundation at all? And if yes, to what extent, and have you met him? And if so, what's he like?
Michael is fantastic. I've been at the foundation for just over a year and a half, and I've been able to meet him twice or three times now. Of course, no one's seen anyone at the foundation in person in about a year, which hasn't helped, but he is absolutely fantastic. I truly mean it when I say that he's everything that he appears to be on TV and in magazines. He's genuine and kind and motivated. Yes, he is involved in the foundation. He, of course, founded it back in 2000, and he is on our board of directors. He doesn't come into the office every day, but he's absolutely involved in the larger strategic decision-making that you see from any nonprofit board. So he's very motivated to stick with us and help us uncover every avenue of treatment that we can possibly think of. In fact, I can share with you all after, but he just spoke with our co-founder, Debbie Brooks, on a webinar this week. So I can send that along to you all. He was discussing his new book, which is all about optimism and his feelings as he, oh, there it is. Jennifer just held it up. And I'm happy to share that with you all. And you should all give the book a read.
Excellent. And we had a question that just came in from Jason. And Jason wants to know, have there been any recent discoveries around repurposed drugs?
So repurposed drugs, as I mentioned before, is a very exciting field for us to work in because we're able to kind of move a lot faster. I don't have any studies off the top of my head that are currently in clinical trials, but we have seen some work in the past few years, and I'm happy to send some more information. One that comes to mind is called nilotinib, and that's a study that we funded a few years ago. And I do want to mention that, as I said before, that trials that don't often go the way we hoped are still very important for our learning process. And so nilotinib didn't go perfectly. It didn't cure Parkinson's, but it did offer a lot of information that we're learning from. So I'll link to that study, and if I can find any others that are widely available, I'll share those as well.
And one of our last questions in the chat is, how did you come to work at the Michael J. Fox Foundation?
Great question. Like I said before, I've been here for about a year and a half. And prior to that, I worked at a large healthcare system in New York City. I got my undergraduate degree in biology. So I knew that I wanted to work in science and healthcare and really looking at like scientific progress for the rest of my life. But I wanted to be able to help people and interact with people and meet people in a way that was meaningful for me. So the foundation, the Michael J. Fox Foundation, really has been the perfect mix of all of those things together, where I can learn about science every day from our researchers who are on staff, who I can just walk down the hall and talk to. And I can also meet people like you all who are out in the community, and I can travel to see you and learn about you and make real human connections, which has been just the most special part of the job. So I love it so much, and I'm very, very lucky and honored to be able to work with you all.
And we have a very engaged group. I'm sorry, Susanna, one last question came in. I know we have 1 minute left, but Roger has an interesting question. Do you know overall approximately how many drug trials that the Michael J. Fox Foundation supports have made it all the way through from that initial discovery phase to FDA approval and ultimately in the hands of patients?
Great question. Yeah, so I spoke about Inbrija before, which is that inhaler for off times, and then Kinmobi, which is that under the tongue apomorphine strip, also for off time. So those are our two kind of golden children of where we funded early clinical research, which then got them approved over the goal line. A lot of studies that we fund are basic biology as well as disease modifying treatments. So looking in the space of slowing or stopping the progression of Parkinson's. And as you know, we haven't seen any of those come to market quite yet. So to my knowledge, Jenny, please correct me if I'm wrong, but it's those two, Kinmobi and Inbrija, but there are a lot more that are kind of heading through the pipeline. I also want to mention that through PPMI, that biomarker study that we're doing, people are able to take that data and use it in their own studies. So we see, while we haven't directly funded a study, we've helped people understand ideas that help them formulate hypotheses for their own studies. So we're working more than just monetarily to encourage research all over the field. So while we may not fund a study, we may have been a part of the process for safety or trial design and things like that.
Excellent. And I think that wraps up our questions. I do want to be conscientious of time, so we are 1 minute over. I will just do one last call for questions in the chat. Okay, and there's just been a shout out to Over Parkinson's, wonderful organization. I'm just thanking everybody for your presentation today and also for their continued support with exercise and speech classes. So
I echo that. Thank you so much to Maria and Over Parkinson's for having us today. It's been a pleasure. And like Jennifer said, you guys are great, great questions. And I'm looking forward to following up with all of you. Anything that you may still have questions about, please don't hesitate to reach out and let me know. Let Maria know. Jennifer, we're all here for you. And I hope you have a great Saturday.
Yeah, so thank you both. That was so informative and so helpful. And you two are lovely humans, and we really appreciate all the work that you're doing and that the Michael J. Fox Foundation is doing. And for everyone that is here today joining the call, I have all of your email addresses, so you will get a recording of this this webinar today. You'll also get a copy of the slides. So if you want to review anything specifically, you can do that. And then all of the links that Jennifer dropped into the chat box, those will also be included for your review. So you have all of the resources that were talked about today. And please feel free to share this with family, friends, people that you think might be interested in learning a little bit more, or anybody that you think would have wanted to be here but for some reason couldn't be here today. Please feel free to share this, because the more people that know about this, the faster we can move along in developing therapies and finding a cure one day. So with that, I'm going to thank everybody for attending. Thank you again, Susanna and Jennifer. You two are wonderful. And everybody have a happy Saturday.
Thank you all.
Thank you. Have a great day.
You too.
Thanks all.